This is an OCR A-level biology video on Unit 3.1, Exchange Surfaces and Breathing. This is the last video I'm making on Unit 3, as I made all others. You can watch them if you want to. So today we're going to be looking at the whole of 3.1, including ventilation in fish, ventilation in insects, spirometers, the mammalian gaseous exchange system, but first of all we're just going to look overall at what makes a good exchange system. Right, so first of all, exchange surfaces and breathing. All organisms need to exchange things with their environment. So they take oxygen, glucose in, and CO2 and urea out. The exchange depends on an organism's surface area to volume, their size and level of activity. So there's size, surface area to volume ratio, the adaptations of these organisms, and their levels of activity that all affect the rate of exchange. Okay, so a good exchange surface is going to have a large surface area. It's going to have thin permeable membranes, a good blood supply. So this good blood supply will help maintain steep concentration gradients. The thin permeable membranes help decrease the diffusion distance needed, and our large surface area provides more space for molecules to pass through, such as like in root hair cells. So first of all, let's look at size. So small organisms, so single-celled organisms, have cytoplasm which is very close to the environment in which they live. Diffusion supplies enough oxygen and nutrients to keep cells alive and active. But multicellular organisms have several layers of cells, therefore any oxygen or nutrients diffusing in from the outside have a longer diffusion pathway. So that's a key word there, diffusion pathway. The larger you are, the longer the diffusion pathway, and therefore the slower diffusion. You cannot rely on it as easily. Then we've got surface area to volume ratio. So small organisms have a small surface area and volume, but their surface area is relatively large compared to their volume. So the surface area is large enough to supply all the cell with sufficient oxygen. As size increases, so volume increases further. Okay, so the larger you are, your volume ends up being bigger than your surface area, but unicellular organisms will have a larger surface area than they do volume. So large organism surface area is relatively small compared with their volume, therefore they have a small surface area to volume, and the rate of diffusion is lower. Adapting, so some organisms can change their surface area by adapting to their environment, such as like flatworms. They adopt a different shape, they're very thin flat bodies, giving it a larger surface area to volume, so they can therefore rely on diffusion as a method for transport. The higher the metabolic activity, the more the need for exchange, so therefore a higher demand for exchange systems. So that is going to lead on to the diagram that's just about to come on the screen now, comparing small organisms to large organisms. So small organisms, they've got a cytoplasm very close to the environment, so diffusion is sufficient. They've got a large surface area to volume ratio, so diffusion is sufficient. And they have a low metabolic activity, so a low demand for fast exchange, so diffusion is sufficient for small organisms. But with large organisms, so multicellular, they've got several layers of cells, so a longer diffusion pathway. Diffusion would be too slow, so it's insufficient. They've got a small surface area to volume ratio. As their volume increases faster, diffusion's insufficient. And a high metabolic activity, so a high demand for gaseous exchange and exchange of all sorts of nutrients within the body. One final thing to look at on this page is something I got examined on recently, and it's how you can work out the surface area and volume of a sphere. So that, what I all know is a poor sphere, but these are the two equations you need to learn. Volume equals 4/3 of pi r cubed, and surface area equals 4 pi r squared. We wasn't given the formula in the exam. I don't know if in the actual exam you'd get the formula, but probs just learn it anyway. If Tom Birdseye's watching this, he's probably laughing. So now let's look at gas exchange in mammals. So we've got these 5 things. Once again, in the recent test I've done the marks, I think it was 6 marker, and you had to mention all of these things. So nasal passages, trachea, bronchioles, alveoli, and the diaphragm. So I'm going to talk you through each one and what they do. So nasal passages have hairs in them, and mucus is secreted, and that mucus, it traps pathogens and dust. Trachea have C-shaped cartilage rings which support the trachea and prevent them from collapse. So as you see, some of these points I put down, trachea, bronchi and bronchioles, have very similar arrangement, but I'm not going to repeat all the points for each single one. They also have ciliated cells which waft mucus, so they're epithelial cells, and within these epithelial cells we have goblet cells which secrete mucus again, and that traps microorganisms. So if we look at bronchi and bronchioles, within them they've got smooth muscle which can dilate and contract. So if you're doing exercise, your bronchi and bronchioles can dilate, allowing more air in, and if they're in an area of pollution where you don't want to be breathing in any pollutants, they can contract and ensure less air gets into the lungs, okay, to protect you. Contraction of the airways is what happens with asthmatics as well. So that smooth muscle contracts. Then they've also got elastic fibres which can undergo elastic recoil, which deals with the intense pressures within the lungs. So when you breathe in and out, the lungs are put under severe pressure, and including these airways, and these elastic fibres allow the trachea, bronchi and bronchioles to recoil and maintain their shape. Then we've got alveoli, which are the main exchange surface. So they've also got this elastic fibre to prevent them from collapsing. They contain surfactant within them, which is a liquid. O2 diffuses into it easily. They've got thin squamous cell walls, and so does the millions of capillaries that surround them. So there's a large surface area of capillaries, and these thin walls create a short diffusion distance. As well as this, capillaries have got very close contact with the alveoli walls, so once again reducing the diffusion distance. Capillaries are also really narrow, so sometimes they're 1 red blood cell thick, which is like, I think, 7 micrometers, pushing the red blood cell against the capillary wall, making them closer to the air in the alveoli. Therefore they have a greater effect on the concentration gradients. They can associate to more oxygen easily, and carbon dioxide readily dissociates. They have a good blood supply overall though, so blood is constantly pumped through the capillaries. It becomes oxygenated, and oxygenated blood leaves. That good blood supply ensures that steep concentration gradients are maintained, so the rate of diffusion is always maximal. Finally, we have the diaphragm and all of the other breathing muscles. So the diaphragm basically changes the volume of the lungs, and it ensures that fresh air is constantly getting into the lungs. So it's once the carbon dioxide has left the blood, you exhale, release all that air containing carbon dioxide, and that's now lowering oxygen, and then you inhale by increasing the volume of the chest cavity, so lowering the pressure. Air's going to rush in, and then you can maintain those high concentration gradients. Okay, so main things overall is you're looking to create a short diffusion distances. You want to get rid of mute agents and microorganisms, anything that will decrease the rate of gaseous exchange that could interfere with alveoli. You want to maintain high concentration gradients. They're basically your three main points, but just learn all these points and you will hopefully do well in the question. This is a diagram of the mammalian gas exchange system, showing you all these points. I'm not going to speak through each of them, it's just showing you the air passages that I've already spoke to here. If you don't understand it, pause the video and just have a look, and each one of them will relate to what I've already spoken to, but it's not anything I need to go into intense detail on. Right, so now we're going to look at spirometers. So as you can see, I've just inserted the picture of a spirometer onto the page, and spirometers basically measure the movement of air in and out of the lungs as a person breathes. So first of all, we've got an oxygen filled chamber with a movable lid. So here we've got our movable lid, and that is attached to a sort of pen onto a chymograph. So when you breathe in and out, the lid moves up and down, moving this pen, and it records a spirometer trace on here, and you can see the little squiggly lines appear on it. Okay, so let's have a look at the stages. So first of all, we've got the person breathes in to our oxygen chamber. So they breathe through a tube connected to the oxygen chamber. As the person breathes in and out, the lid moves up and down. Movements can be recorded by a pen attached to the lid, which writes on a rotating drum called the Spirometer Trace. So this drum rotates around, the pen moves up and down, you record the trace, or an electronic signal can be produced using motion sensors, but you can just remember it as this trace. We've got soda lime in here, which absorbs carbon dioxide. So basically we know that the gas in here is oxygen. Okay, all the carbon dioxide is consumed, so we know that we're breathing out oxygen in this chamber, so it's measuring the amount of oxygen in and out. The total volume of gas decreases over time in this spirometer as oxygen is inhaled and CO2 is absorbed by soda lime. So here I've just drawn this graph with 3 labels on it. I'll just get the third label out, which is residual volume. Okay, so when we're looking at this graph, as you can see, the normal waves are our tidal volume. So our tidal volume is the volume of air moved in and out in each breath, and they're going to be the short waves on our graph. Then we've got this large wave, so it's your maximal air inhaled followed by the maximal amount of air exhaled, which is your vital capacity. So you breathe in as much as you can and then exhale as much as you can. So the literal definition is the max volume of air moved by the lungs in one breath. Okay, so as much as you can in and as much as you can out. And the final thing you need to know is residual volume, which is a volume of air that remains in the lungs, the minimum volume of air remaining in the lungs at all times. Okay, so you'll never have less air than this residual volume, because if you don't breathe out all that air in the lungs, otherwise you'd get such low pressure your lungs would just collapse in on themselves. You need some sort of pressure to maintain the lung shape. Okay, from this graph you can work out your breathing rate, so you can measure the peaks in the tidal volume per minute. Height, age, gender and physical fitness can affect your vital capacity. So that was spirometers. Now let's look at ventilation in fish. So we're looking at bony fish when we're talking about that. This is what OCR want us to learn. So let's just think about the situation first. So there's low O2 levels in water, much lower than in airs. Oxygen does not really readily diffuse into water that easily. Most fish have 5 pairs of gills covered by a bony plate called the operculum. Each gill consists of 2 rows of gill filaments attached to a bony arch. Filaments are thin and folded into gill plates called secondary lamellae that provide a large surface area. Okay. Exchange takes place between the blood and secondary lamellae. So hopefully now a diagram of the fish is going to come on. I'm speaking too fast. Right, here is our diagram. So first of all, we're going to look at the bottom here and sort of ventilation. So as before, we looked at breathing in and out in the lungs. Now we're going to look at how water carrying oxygen moves past our secondary lamellae. Okay, so how it gets in and how it's exhaled as such. So first of all, the fish opens its mouth. So here, the inside of the mouth we call the buccal cavity. We're dealing with pressure differences here. You open the mouth, lowering the floor of the buccal cavity, and the pressure inside the cavity therefore decreases. So the fish opens the mouth, the pressure decreases as the floor lowers, therefore water rushes in. Then you close the mouth. This increases the pressure within the buccal cavity, but the mouth is closed as the buccal cavity floor rises. So water is forced down here past our gill arches and secondary lamellae, and the pressure that then opens our operculum for the water to exit. So water comes in here, opens the mouth, buccal cavity lowers, water rushes in, mouth closes, pressure rises, and water is forced through past the gills and out of the operculum. Now let's look at countercurrent flow in fish, which is represented here. So water flows across our gill arches, okay, opposite to the direction of blood flow. This is called countercurrent flow. Blood flows along the gill arch, along filaments of secondary lamellae. Blood flows in the opposite direction through capillaries to the flow of water. The concentration of oxygen is always higher in the water than the blood, and fresh blood is constantly exposed to the water. Okay, so by moving blood across water, then fresh water with new oxygen arrives, and that oxygen is transferred to the gill arches, and it maintains our concentration gradients, which basically what this graph is showing. I won't go into too much detail on that graph. It's probably in your textbooks and you can look at it yourself. Now, finally, let's look at have completely different sort of ventilation to both mammals and fish. So they, as we know, they've got open circulatory systems and an air-filled tracheal system. So air enters the insect via spiracles. O2 travels down the concentration gradient towards cells. CO2 travels down the concentration gradient towards the spiracle. So O2 and CO2 flow in opposite directions. Air is transported through the trachea to the tracheoles to the tissue fluid. Okay, so a diagram should come up around about now, and it's going to show you the passage of air. So air enters in our spiracle outside the insect. It flows through the trachea, tracheoles, and here we've got tracheal fluid, and then oxygen diffuses into the tracheal fluid, and the tracheal fluid carries that oxygen into the cells, into the tissue fluid, okay? Tracheal fluid is also called tissue fluid. So tissue fluid in the trachea is withdrawn into the body fluid to increase the amount of tracheal space exposed to air. Okay, so one thing I didn't show you here is we've got rings of chitin to support the vessels, just like we have cartilage in our trachea, in the lungs. So this diagram is showing you tracheoles. So this is tracheal with blue cells on the outside, and inside it we've got tissue fluid, which oxygen diffuses into and waste diffuses out, so carbon dioxide diffuses out of there. The insect can withdraw some tissue fluid, putting it into the body fluid to increase the amount of tracheal exposed to the air, so more oxygen is then absorbed by these cells and more gets into the insect. Okay, a fairly easy concept to understand. It's pretty simple, but they struggle to change the volume of these, and they're pretty much relying on diffusion at this rate. So let's look at how insects can alter the volumes and the pressure within their trachea. So sections of the tracheal system are expanded and have flexible walls acting as air sacs, which can be squeezed by action of flight muscles. Okay, movement of wings can alter the volume of the thorax. As the thorax volume decreases, the pressure increases, therefore air is pushed out. If thorax volume increases, there is a decrease in pressure and air moves in. Valves in spiracles can be opened and closed within the thorax, causing air to flow from the thorax to the abdomen. Okay, so to sort of represent that on the diagram, what I've just splurged out to you, at the moment, if we can pretend this is our thorax, which it's probably not, just pretend it is. So at the moment it's quite wide. What the insect can do is, by flying, by contracting this thorax, it can decrease the size of the thorax. So let's forget this bit here now. So that's going to increase our pressure and therefore squeeze air out of here. Okay, so air is going to leave. If the insect relaxes the thorax, so the thorax volume increases, and we go back to the previous shape, then we've got a decrease in pressure and air is going to flow in. There's one way insects can do it. Other ways, they've got valves which can be opened and closed. So that valve is closed and now it can be opened, okay, to let air flow in and out and control to which parts of the body air is moving to. So you might get a question why insects would need to fly or constantly be need to moving, and that is because it alters the volumes within their thorax and therefore alters the pressure within their trachea to control the movement of air in and out. That was all on Unit 3.1. Thanks for watching.